everyone, my name is Megan Collins. I'm the research coordinator for CureGRIN, and today I am joined by Dr. Graham Collingridge. Dr. Collingridge, could you please introduce yourself?
Yes, good afternoon. So my name is Graham Collingridge. I'm the director of the TAN Centre for Research into Neurodegenerative Diseases at the University of Toronto. I'm also a senior investigator at the Lumenfeld Tannenbaum Research Institute at Mount Sinai Hospital in Toronto.
Wonderful. Thanks so much for joining us today, Dr. Collingridge. So our first question is, what are the key takeaways from your research?
Well, we're interested in understanding how the brain stores information, and we focus on the communication at the level of synapses, which are the points of connection between neurons. And over the years, we have identified, firstly, the role and many of the mechanisms by which a specific protein, the NMDA receptor, is involved in these processes. So going back to when I was a postdoc in Vancouver, we discovered that the NMDA receptor is the trigger for the process known as long-term potentiation, or LTP, which is the widely studied model for understanding how the brain stores information, how it learns and remembers, etc.
Perfect, thank you. So our next question is, what questions will your research help answer about GRIN disorder?
Well, I think it will answer two main questions. The first is a better understanding of how the NMDA receptor functions normally. I mean, we know quite a bit now. I mean, there's many groups around the world been working on the NMDA receptor, and we have a very good understanding of much of what it does, but there's still information that we don't know. There's still things we don't know about the NMDA receptor normally. But more specifically, by studying variants that are observed in human subjects, we should be able to understand what is actually causing the changes specifically related to the point mutations that are being observed. So we're studying, for example, the G620R variant of the GRIN1 gene, and we already have quite a number of differences between the mouse models that we have, which express this variant compared to their wild type littermates.
Perfect, thank you. So our last question is, what are you most excited about for your lab in the future?
I think there's two things in terms of the work we're doing in the GRIN project, which actually is my favorite project by a long way. The first is to see whether we can restore function of the NMDA receptor using positive allosteric modulators. These are drugs which will boost the function of NMDA receptors. And we know that we've got roughly a 50% reduction in normal function in our mouse models. So given an effective PAM, as they're called, we would hope to be able to restore that function, and then we can see whether we can reverse some of the behavioral deficits that we also observe in our mouse models. In the long term, I'm very keen to form part of a collaboration to see whether it's possible to get a genetic rescue. This would be really exciting, either by overexpressing the normal gene or eventually doing a genetic editing rescue to remove the variant and replace it with the normal gene. But these will be part of a collaborative study led by Amy Ramsey and others around Toronto and throughout the world.
Wonderful. Well, thank you so much, Dr. Collingridge. This has been super exciting. Thank you for your time and for sharing with us today.
You're very welcome.